Alright guys, so last time we ended up talking about carbohydrates. This is where we started off. Last time we talked about lipids and how those were different fats, how there were three different kinds of lipids. There were phospholipids, which were in the cell membrane, saturated and unsaturated fats. Those are going to be your grease and oil. And then finally we talked about how there were steroids and cholesterol, how those kind of looked like chicken wire, things like that. Sorry if this video sounds different than my other videos. I'm recording this one at home on my home computer, so I'm not sure how the mic is going to really do with that. So, carbohydrates. Carbohydrates are what the cell uses for short-term energy. That's why a lot of coaches will have you quote unquote carb up before a big race. Gives you just a little bit of extra short-term energy. You can think about what happens when you eat something as well. So when you eat something, your blood sugar increases, so the amount of carbohydrates in your system increases, and it gives you kind of a short-term boost. We also use it for structural support in a couple of different ways, which we will talk about. Some of the distinguishing characteristics is carbohydrates are made of carbon, hydrogen, and oxygen. And they always follow in the same exact ratio. So for every one carbon, there are two oxygens, and for every one carbon, there are two hydrogens and one oxygen. So here we have 6 carbons, which means we have 6 oxygens, and 12 or 2 times the number of hydrogens. So it's kind of a cool distinguishing characteristic of that. So there are 3 main types of carbohydrates: monosaccharides, disaccharides, and polysaccharides. And I think we talked about this last time, so that would be block day. I think we talked about this on block day. But mono means just one, whereas poly means many. So what I've done here is you can see this is just one monomer. So this is a simple sugar. This is actually the molecule glucose, which is one of the important ones. The formula for glucose, which I would like you to memorize, is going to be C6H12O6. And that's going to come up a bunch throughout the semester, so you will be seeing that again for sure. Another one is ribose, which we'll be talking about a lot next unit. Ribose is C5H10O5, and you can see once again it follows that 1 to 2 to 1 carbon hydrogen oxygen ratio. So disaccharide is a form of polysaccharide, is a little bit more specific. So you can see down here there's more than one, so it's not going to be a monosaccharide, but there's only two. Di is the prefix for two, so disaccharide is just 2 of them. So two really common ones that we talk about a lot: sucrose, which is just table sugar, and lactose, which is a combination of glucose, this guy up here, and galactose. So not being able to break these two things apart is what makes you lactose intolerant. So you can't break these two molecules apart. So polysaccharides contain 3 or more. Generally, when we're talking about polysaccharides, we're talking about thousands of sugars linked together. What these generally look like is they look almost like a chain. So you have each individual link of the chain, that's going to be one monosaccharide, and then the whole polysaccharide is going to be the whole chain. Big ones that we talk about here, we talk about starch. Starch is the plant glucose. Starch is going to be found in things like potatoes, so lots of vegetables. Glycogen, these are our animal glucose chains. Glycogen is how we store blood sugar. So our blood sugar is going to be mostly in the form of glycogen, and hopefully we'll get to talk about that when I get back on Tuesday, because we'll be hopefully talking about feedback loops and things. So cellulose is plant fibers. These are the cell walls. We as humans and as animals can't really break down cellulose. That's what we call fiber. And then chitin is the exoskeletons of things like spiders or shrimp or crustaceans, anything that has that hard exoskeleton. Bugs have chitin shells. And then fungi, which is kind of weird, but that's just one kind of cool thing that they have that makes them a little bit different than regular plants. Alright, so nucleic acids, we will be talking about these so much next unit, so I'm just going to kind of gloss over these real quick. So there's two main types of nucleic acid. There's DNA, which are deoxyribonucleic acids, and then there's RNA, which are ribonucleic acids. And you can kind of see right off the top, if you remember ribo, that was something that we talked about when we were talking about sugars a second ago. Ribose is the sugar that's used in RNA, whereas deoxyribose is used in DNA. So it's just one difference between them. But every monomer of all nucleic acids are made up of a 5-carbon sugar. In case of DNA, it's deoxyribose. RNA, it's ribose. So you have that sugar right here. Attached to the sugar, we have a nitrogenous base. So this is an amine. Remember, an amine is anything that has this NH2 functional group up there. But we call this a nitrogenous base. There are 4 different bases. The sequence of those bases makes up the DNA sequence. And then we have our trusty phosphate group, which we also learned about. So that's kind of it. No, didn't mean to do that. So that's kind of it for nucleic acids. So moving on to proteins. Proteins are chains of amino acids. So the monomer of a protein is an amino acid. The monomer is an amino acid, and the polymer we will sometimes call a polypeptide. Anytime you hear the word pep, that's going to be referring to protein in some way. So we call them polypeptides, or we call them proteins. So the job and the function of these, they serve as structural components. They can move things around. Enzymes, big one. I'm actually going to have you watch the presentation I made for my Biology 1 kids on this. It was a pretty good one, and I think you guys will like that. And then cell chemosignals, we use proteins to talk to each other. And I'm going to come back to this picture here in a minute because it doesn't fit on the slide that I actually talk about this stuff on, but I will be coming back to this photo. All right, so the amino acids, and I think it's going to get cut off here somewhere on the edge. So amino acids have a carbon center surrounded by an amino group. So we talked about that amino group, NH2 on the end, our center carbon, and a carboxyl group. Remember, it's carboxyl because this is not an R group, it's a hydroxyl group. A hydrogen and an R group. And a lot of people yesterday were talking about what R groups are. So R groups are just something else that's stuck on here. So we can take a look over here at all of our different amino acids. These are all the amino acids there are. And you can see that up here that's not in the box is just this. It's the same exact thing. All amino acids are the same up top. But then if you look here, they have something different attached to the bottom of all of them. That's what an R group is. It's just something else. I think this is a good kind of example to really see what an R group is. So those R groups make all of those amino acids different. So you can see that some are acidic, some are basic, some like water and are polar, some do not like water, they're non-polar. So there's kind of all different kinds of proteins. And that really has to do with what the R group is. All right, so this is the slide that I'm going to go back to the picture on in just a second. There's 4 different levels of protein structure. There's the primary structure, secondary structure, tertiary structure, and quaternary structure. So the primary structure is just the order of the amino acids. So we had all of those different amino acids that can combined in any order. So the primary structure is just what order are they in. Your secondary structure is the actual 3D arrangement of the proteins caused by hydrogen bonds. So they kind of scrunch together and they form hydrogen bonds together. Tertiary structure is when you start getting into this 3D structure caused by the interaction of the R groups. If you'll remember when we were talking about the sulfhydryl group, I said that it is influential in protein folding. This is why. One of those R groups is a sulfhydryl group, and it specifically folds the protein into a very, very specific structure. And then quaternary structure, not all proteins are going to have a quaternary structure, but essentially what a quaternary structure is it is individual protein subunits working together to make a big protein. So that's what it is. Now I'm going to go back to the other picture real quick. So here we can see our primary sequence, so it's just them strung together. So that's going to be the primary sequence. And then here we start seeing that secondary structure. So there's some bonds forming here. The R groups kind of come in this tertiary structure and make it this wonky looking thing here. And then for the quaternary structure, you can see that there's one protein here, one here, one here, and one here. But they all work together to make hemoglobin. So it's 4 individual proteins working together to make one quaternary structure protein. All right, so enzymes. And I did a much better job teaching enzymes to my Biology 1 kids, so I'm going to link that video below this one. Please go through, take a look at it. You don't have to take notes on it, but kind of go through, look at it, see what I have to say in there. And a lot of the stuff's going to be really similar to this. So proteins act as a catalyst. What a catalyst does is it speeds up reactions by lowering the activation energy, but it doesn't directly take part in the reaction. One more key vocab word that you need to know is what substrate is. So a substrate is whatever, oh sorry, y'all have told me this really late. So a substrate is the substance that an enzyme acts upon. So it's whatever is going into the reaction. So without an enzyme, you can see that we just kind of don't have the energy to quite fully start the reactions. We can't get from substrate to product. With an enzyme, essentially what we do is we lower the amount of energy that it takes to start a reaction just in general. So there's a couple of different parts of an enzyme. There's the active site. The active site is the part of the enzyme that interacts with a substrate. The active site is created by the 3D shape of the protein. And again, there's a video of me actually holding up examples of enzymes and things in the Biology 1. So please go through, take a look at that. I will link that below. Induced fit model, essentially what this is it describes the action of the substrate fitting into the active site of the enzyme. The enzymes function best at their optimal values, but their effectiveness can be affected by temperature, pH, concentration of both the enzyme and the substrate. The way I kind of think about it is I don't want to be running around. I'm not very effective when it's 10,000 degrees outside. Enzymes don't work that way either. They want specific temperature requirements. So this is kind of what it looks like when we have our substrates fitting together. So our substrates are whatever's going to be affected. You can see that it fits right into the enzyme. It fits perfectly in. The enzyme helps the reaction to happen, and then it goes away. So as we're watching this, our substrates fit perfectly into the enzyme's active site, which is down here. Something's happening here, so the enzyme's bringing them together, and then the product releases. But you notice the enzyme stays exactly the same throughout the whole thing. Enzymes can be reused again and again and again and again. All right, so inhibitors. There's 2 types of inhibitors. There are the competitive inhibitors that resemble the substrate and block the active site directly. So you can kind of think of jamming a key into a lock that isn't the exact key and then breaking it off. So it's directly competing for where you put the key in. And a non-competitive inhibitor changes the shape of the enzyme so it no longer functions. So you can think of almost putting silly putty over the key, and so it's not going to really work well. So that is it. I would really like you guys to go through and look at the enzymes PowerPoint that I have up on Schoology right below this one. It goes way more into depth. I did a much better job explaining it in that one. So I do want you guys to watch that. If you think that's going to help you, please take notes off of that. And I hope I will see you guys Tuesday. Please have a really safe weekend. Remember Miss Williams' #1 rule, don't do anything to end up on the paper or the internet. All right, have a safe weekend, guys.